everyone, my name is Carly. Today I will be lecturing on the biosynthesis of amino acids, which is the key building blocks to proteins. So let's get started. So amino acids can either be categorized as essential, non-essential, or conditionally essential amino acids. So essential amino acids are those that cannot be made by the human body and therefore must come from the diet. Non-essential amino acids are those that can be made by the body, so we possess the correct biosynthetic pathways in order to synthesize these amino acids. Conditionally essential amino acids are those that are essential only under unique physiological conditions, such as pregnancy and adolescence. So these amino acids, we do possess the biosynthetic pathways for. However, they cannot be produced under special circumstances. So here is a graph showing the number of steps in the pathway to produce non-essential and essential amino acids. As you can see, most non-essential amino acids require few steps, and most essential amino acids require many steps in order to produce. So why is that? Because the more steps that are required to produce an amino acid, the more enzymes we need to possess and produce in order to make this amino acid. So if we can acquire these difficult to acquire amino acids through our diet instead of synthesizing them de novo, then it will make us much more energy efficient. So, which is why over time we have evolved to not have these amino acids, these enzymes to produce these essential amino acids, and instead require these essential enzymes in our diet. So these are the amino acids that are essential and non-essential in humans. However, it's a different story for bacteria. So different bacterial strains have different amino acid biosynthetic pathways. And there are two terms we're going to go over. You've probably seen this in your earlier classes, but we're going to go over them today. So a prototrope is a strain that's capable of producing all its metabolites from inorganic material. So what this means is if you grow a prototype in minimal media, it will still grow because it can produce 20 of its amino acids because it possesses all the different amino acid biosynthetic pathways. However, an oxiotrope is a mutant strain that requires a particular organic compound in order to grow. So, for example, here we have an arginine oxiotrope that requires arginine in its media in order to grow. So this oxiotrope cannot grow in minimal media because it cannot synthesize arginine. Therefore, it cannot survive without arginine in its media. So, however, it can grow in minimal media that is supplemented with arginine. So here's another example of this. So here, histidine converts histol into histidine. So if you grow the histidine possessing strain and the histidine negative strain in media containing histidine, then both strains would grow. However, if you grow them in histidinol, then only the strain that contains histidine can grow because it converts histol into histidine. However, the strain without histidine cannot grow because it cannot do this conversion, and therefore it cannot acquire histidine. Now here are the overviews of the biosynthetic pathways of all the amino acids. And these are the pathways that we humans possess. And we will go over all of this in a bit. So before we go over those pathways, I'm going to go over the transaminase reaction, which is a very important reaction in amino acid metabolism. So this transaminase reaction transfers a amine group from a amino acid to a keto acid in order to produce a different keto acid in a different amino acid. So this transaminase reaction requires a PLP cofactor in order to occur. So this will be, we will go over the mechanism of this in further detail a bit later. So this transaminase reaction is usually described as a ping-pong mechanism. So why is that? It's because so in the reaction mechanism, the first substrate enters the enzyme, and then the first product is produced and released out, and then the second substrate binds, and the second product is released. So for aspartate transaminase, you can see that the amino group in the aspartate is transferred to the alpha-ketoglutarate, producing oxaloacetate and glutamate. So the mechanism here is shown. So the first substrate, the aspartate, binds onto the PLP cofactor. And eventually, for a series of reactions, the first product, oxaloacetate, is released, which leaves the amino group attached to the PLP cofactor. And then this amino group will be transferred onto the second substrate that binds, which is the alpha-ketoglutarate in this case, and the second product will be made and released, which is the glutamate. And this is in the second half of the mechanism, which is not shown here. Now, another transaminase reaction is the alanine transaminase reaction, which transfers the amino group from alanine to alpha-ketoglutarate to make pyruvate and glutamate. And this is a branched chain amino acid transaminase reaction, which converts a branched chain amino acid into a keto acid by transferring the amine group from the branched chain amino acid to an alpha-ketoglutarate to form glutamate and a unique keto acid. So now we're going to go over all the biosynthetic reactions in humans. So many amino acids are derived from alpha-ketoglutarate, including glutamate, glutamine, proline, and arginine. So for arginine biosynthesis, we'll go over this in the next lecture during the urea cycle lecture. So it won't be shown here. Now for glutamate Now, alpha-ketoglutarate can be converted to glutamate in one step, either through the transaminase reaction, which we have seen before, or the glutamate dehydrogenase reaction, which uses up a ammonium and either a NADH or a NADPH. Now, NADPH is actually an important cofactor used for many biosynthetic reactions, including in amino acid biosynthesis. So the difference between NADH and NADPH is a simple phosphate group on here, which is why there's an extra P in the name. Now, glutamine biosynthesis can occur in one step by converting glutamate into glutamine. So glutamate can first be activated using ATP in order to add that phosphate group on there. And then this activated intermediate will be attacked by ammonia, which gets rid of the phosphate and adds the ammonia on there, making glutamine. And this reaction is catalyzed by glutamine synthase. Note that synthase is different from synthase. So synthase is an enzyme that catalyzes the production of a compound. And synthesase is a enzyme that catalyzes the ligation of two compounds together. In this case, that's the glutamate and the ammonia. Now, glutamate can also be converted proline after many more steps. So glutamate is first activated by ATP via glutamate kinase, and then this intermediate can be converted into glutamate semialdehyde using NADPH. Then this intermediate can spontaneously form the ring in proline via a simple dehydration reaction. Then the 5-PC can be reduced by NADPH to produce our ultimate compound of proline. Now we're going to go over the amino acids that are synthesized from oxaloacetate. So oxaloacetate can be made from aspartate and asparagine can be made from oxaloacetate. So oxaloacetate can be converted to aspartate via the aspartate transaminase reaction seen previously, and then aspartate can be converted to asparagine via asparagine synthesase, which is a glutamine aminotransferase reaction. This reaction releases an ammonia from the amino group of glutamine and transfers it onto aspartate. So note that this type of reaction is not the same as the transaminase reaction. It uses a very different mechanism. So in this reaction, in the asparagine synthesase reaction, glutamine enters the enzyme in the glutamine binding domain and forms a thioester bond with the cysteine that is part of the enzyme. And consequently, the ammonia is released from the glutamine. And once released into the enzyme, the ammonia travels to the ammonia acceptor domain and attacks the activated substrate. In this case, it's the AMP aspartate intermediate to form asparagine. And then the glutamate that's attached here is released by a simple hydrolysis reaction to form glutamate. Now we're going to go over the amino acids that are synthesized from free phosphoglycerate here. So serine can be produced from free phosphoglycerate. First, free phosphoglycerate is oxidized by NAD plus into this intermediate, and then this intermediate undergoes a transaminase reaction, which we haven't seen in the previous transaminase slides, into free phosphoserine, and then this intermediate it undergoes a simple hydrolysis reaction in order to produce serine. Now onto glycine biosynthesis. So here serine reacts with THF and transfers a methylene group onto THF in order to produce glycine and methylene THF. So interestingly, methylene THF is a great methyl donor in DNA metabolism and can convert UMP to TMP. And this reaction actually recycles the methylene THF back into THF for glycine biosynthesis. And this pathway is actually very highly active in cancer cells, which we will discuss in a later lecture. As for cysteine biosynthesis, it begins with methionine being converted into SAM. Then SAM has its methyl group removed, and then the adenosine group removed to form homocysteine, and this is combined with a serine molecule in order to produce this intermediate. Then this intermediate is hydrolyzed and deaminated to form cysteine and another substrate. So now the last amino acid we're going to go over in human biosynthetic reactions is tyrosine. So tyrosine is actually not formed via the typical aromatic amino acid biosynthetic pathways. Tyrosine is synthesized by phenylalanine in humans, and the phenylalanine is acquired via our diets. So the phenylalanine from our diet is converted to tyrosine via using oxygen and THBP. So during this reaction, THBP is oxidized into DHBP. And then this DHBP can be regenerated back into THBP via a reduction reaction using NADH. And this phenylalanine to tyrosine reaction is catalyzed by phenylalanine hydroxylase. And if you have a deficiency in this enzyme, you would actually have a metabolic disorder called phenylketonuria, PKU, which means that you would have a buildup of phenylalanine in your body. And actually the treatment for, the treatment for this disorder is to decrease the levels of phenylalanine you obtain from your diet. Now we're going to go over the biosynthetic pathways in bacteria and plants. Bacteria and plants can also use other biosynthetic pathways that we do not possess. So one of these pathways is the GS-GO-GAT cycle. So we actually do produce this glutamine synthesis reaction as seen previously that converts glutamate into glutamine. But bacteria and photosynthetic eukaryotes can convert glutamine back into glutamate by converting an alpha-ketoglutarate to a glutamate and you consuming an NADPH. And bacteria can also have the aromatic amino acid biosynthetic pathways. So this begins with a PEP from glycolysis and a eros 4-phosphate compound from PPP. And by using these two compounds, via multiple reactions, you eventually get an important intermediate for aromatic amino acid biosynthesis called chorismate. This chorismate can be converted to a phenylalanine and tyrosine, and also tryptophan here. So chorismate goes through several biosynthetic reactions in order to reach to be converted into tryptophan, but some of the key features is that the PRP is used up in the reaction. PRP is actually the main precursor to purine biosynthesis, which we will learn later. And this series of reaction produces GAP, which can then go into glycolysis. And it uses up serine in this last step. Now for lysine, methionine, and threonine biosynthesis, it begins with aspartate. So aspartate can be converted into either lysine, threonine, or methionine. As for branched chain amino acid biosynthesis, it begins with threonine, which was produced by aspartate, as seen previously. This threonine can be converted into alpha-ketoglutarate, and this compound can be combined with pyruvate in order to produce isoleucine. Or two pyruvates can be combined to produce leucine or valine. So that's it for this lecture. So some learning objectives for this lecture is know your essential and non-essential amino acids, know how they work in humans and how they work in bacteria, and understand transaminase reactions, and also know all the amino acid biosynthetic pathways that we went over in both humans and bacteria and plants.